Hi everyone, welcome to my channel. Today I will discuss about interview questions and answers for the stability section in Pharma. What is pharmaceutical stability? Stability in pharmaceuticals refers to the ability of a drug substance or drug product to maintain its identity, strength, quality, and purity throughout its shelf life. Why is stability testing important? Stability testing ensures that a drug remains safe and effective over its intended shelf life under specific storage conditions. What are the different types of stability testing? Stability testing types include long-term, accelerated, intermediate, stress, and in-use stability testing. What is ICH and how does it relate to stability testing? The International Council for Harmonization provides guidelines for stability testing. Example, ICH Q1, Q1F, to ensure global harmonization. What are the standard storage conditions for stability studies? Common conditions, long-term, 25 degrees Celsius plus or minus 2 degrees Celsius / 60% RH plus or minus 5% RH, accelerated, 40 degrees Celsius plus or minus 2 degrees Celsius / 75% RH plus or minus 5% RH, intermediate, 30 degrees Celsius plus or minus 2 degrees Celsius / 65% RH plus or minus 5% RH. What is a stability protocol? A stability protocol is a detailed plan that outlines the design and execution of stability studies, including test parameters, storage conditions, time points, and acceptance criteria. What is the significance of bracketing and matrixing in stability testing? Bracketing reduces testing by evaluating extremes of design, while matrixing reduces testing by testing a subset of samples at each time point. How are stability samples selected for testing? Samples are selected based on the manufacturing batch, pilot or production scale, packaging configuration, and intended storage conditions. What tests are commonly performed in stability studies? Typical tests include assay, degradation products, dissolution, moisture content, pH, and physical appearance. What is photostability testing, and why is it performed? Photostability testing evaluates the effect of light exposure on a drug product or substance, ensuring it remains stable under light conditions. What are the key ICH guidelines for stability testing? Key guidelines include ICH Q1A, stability testing of new drug substances and products, ICH Q1B, photostability testing, ICH Q1C, Q1F, variations and special cases. How are stability study results reported? Results are compiled in a stability report, summarizing test data, trends, and conclusions, and submitted to regulatory authorities as part of the drug application. What is shelf life, and how is it determined? Shelf life is the period during which a drug remains within specifications. It is determined through statistical analysis of stability data. What is a stability indicating method? A stability indicating method is a validated analytical method capable of detecting and quantifying the degradation products while ensuring the active ingredient remains measurable. How do you handle out-of-specification results in stability studies? OOS results are investigated according to SOPs, including root cause analysis, impact assessment, and corrective actions. What factors influence the stability of a pharmaceutical product? Factors include temperature, humidity, light, pH, packaging, and interaction between ingredients. What is force degradation, and why is it performed? Force degradation involves exposing a drug to extreme conditions, example high heat, acid/base, oxidation, to identify degradation pathways and validate stability indicating methods. What challenges arise in stability testing of biological products? Challenges include sensitivity to environmental factors, complex formulations, and shorter shelf lives compared to small molecules. How do you determine the retest period for drug substances? Retest periods are determined based on stability data, indicating the period within which the substance remains within specifications after manufacture. What are the criteria for selecting packaging materials for stability studies? Packaging materials are selected based on compatibility, protection against environmental factors, and regulatory requirements. What would you do if the assay value drops below specification at an intermediate time point? Investigate for trends, check environmental conditions, evaluate method accuracy, and assess batch quality. Corrective actions may include resampling or extending studies. How would you approach stability studies for a new combination drug product? Evaluate the stability of individual APIs, assess interactions, and conduct combination-specific stability studies under ICH conditions. How do you design a stability study for a product intended for tropical climates? Design studies under Zone 4 conditions, 30 degrees Celsius per 75% RH. As recommended by ICH guidelines, what steps are taken when a formulation fails? Accelerated stability testing, analyze failure modes. Reformulate if needed, and conduct stress tests to identify weak points. How do you manage stability data for lifecycle management of a product? Use stability data to support post-approval changes, shelf life extensions, and ensure ongoing compliance with regulatory requirements. Thank you for watching.